protects the cell. Sometimes some eukaryotic bacteria, they have cell wall, like fungi. So fungi, yes, they have cell wall. About prokaryotic cells, most prokaryotic cells, they have cell wall, they have a complex, they have, a structurally complex cell wall, I would say. And we can actually classify bacteria based on the structure of their cell wall. We will be talking more about the cell wall, which protects the bacterial cells. So just remember for now that most bacteria, yes, they have cell wall, but there are exceptions. As I told you, Mycoplasma is a bacterium which doesn't have a cell wall. So when it comes to reproduction, eukaryotic cells, they reproduce both sexually and asexually, while prokaryotic cells, including bacteria, they reproduce only asexually. And also we will be talking about this type of reproduction in bacteria in the next session. Some eukaryotic and some prokaryotic cells, they are able to move. For movement, they need flagellum. Also, I will be telling you more about flagellum. So it doesn't mean that all types of eukaryotic and all types of prokaryotic cells, they have flagellum. That's not true. Some of them, they have flagellum, and flagellum help the cells to move. And also we have electron transport chain in eukaryotic cells, as I told you, which reside in mitochondria. So mitochondria, as you already know, is responsible for generating ATP. You already know that prokaryotic cells, they don't have mitochondria. And electron transport chain if it's present, it resides in the cytoplasmic membrane. But there are, some prokaryotic cells which don't have the electron transport chain like at all. Okay, we will be talking about those bacteria too. So we have a naming system to name organisms, including microorganisms and bacteria. This naming system is called binomial or two-name system. And in this naming system, each microorganism has a scientific name, which has two parts. The first part, or the first name in the scientific name, is the genus name. For example, we have Escherichia coli here, which is a type of bacterium. It is a very famous bacterium. I don't know, but I think that you've heard the name of this bacterium, Escherichia coli. So Escherichia coli is a scientific name. It's a bacterial name, so we have a bacterium named Escherichia coli. And its name has two parts, like the name of other, bacteria. It has two parts. So the first part refers to the genus name, and the second part, which is coli, refers to the species name. So Escherichia is the genus name of this bacterium, and coli is the species name of this bacterium. And as you can see, both of these names, both Escherichia and coli, they are italicized. It seems that the letters are, leaning to the right. So if you want to type the scientific name, you should type it in this way. They need to be italicized. And between the genus name and the species name, there is this space. Also, you should know that the genus name, the first letter of the genus name needs to be capitalized. So E in Escherichia is a capital E. But the first letter of the species name, it must not be capitalized. So as you can see, we have this small c, not the capital C. So these are basically the rules that you need to follow if you want to type or write down a scientific name. Escherichia coli, again, is a scientific name. So when you are writing an essay or something, and if you are referring to a scientific name for several times, just remember for the first time, we need to write it completely, like Escherichia coli. But for the second one and after that, you need to write the abbreviation form of the scientific name, which is E. coli here. So E. coli is the abbreviation of Escherichia coli. So for the abbreviation, first of all, you just need to write down the first letter of the genus name, which is E here. It is followed by a dot. So instead of writing or typing the complete form of the genus name, you just need to write or type the first letter of the genus name, which is E, which is followed by a dot. And then we have this space. And then you need to write or type the species name completely. So for the abbreviation, we write the first letter of the genus name followed by a dot or period and a space, then the species name. So these are basically the rules that you need to follow to write or type the abbreviation form of the scientific name. Again, for the abbreviation, make sure that it's italicized. So we're able to classify bacteria. And in this slide, you can see that we classify bacteria by using several criteria. First of all, for classification, we consider their microscopic appearances. Bacteria are very, very tiny. We can see them with the naked eye. So if you want to see bacteria, you need to see them under the microscope. And this is called the microscopic appearance of the bacteria. So if you want to see how bacteria look when it comes to their cells, you need to see them under the microscope. Some bacteria are spherical, some bacteria are rod-shaped. Okay, so different bacteria have different shapes, and you can basically classify bacteria based on their microscopic appearances, based on what you see under the microscope. But remember that we are able to grow most bacteria in the microbiology laboratory. We are able to propagate bacteria in large numbers, and when we have a large number of bacteria, we can actually see the microscopic appearances of the bacteria cells. So in this slide, you're seeing some bacteria which have been growing in the microbiology laboratory. So again, we can see bacteria with the naked eye. We can see individual cells because we are very, very tiny, and we can see them with a naked eye. But if you grow them in the laboratory, if you have a large number of them, yes, you can see them microscopically. So based on the appearances of bacteria, we can actually see that some bacteria, when we grow them in the laboratory, they have different colors. Even some bacteria, when we grow them in large numbers, uh, they they have this special smell. They can have different sizes, different shapes. In the left image, you're seeing bacteria which have been growing on this type of culture medium in the laboratory. And you're seeing isolated colonies. Each colony that you see in this type of culture medium is the result of growing only one bacterial cell. So each isolated colony that you are seeing in these culture media, they are resulted from growing only one bacterial cell. So you can actually compare the bacterial colonies with each other. Sometimes you can actually classify bacteria based on the appearances of their colonies, based on their color, based on their size. Some bacterial colonies are very large, some bacterial colonies are very small. They can have different shapes, and also, as I said, sometimes when you grow bacteria in large numbers, they have this special smell. You will be working with bacteria in the microbiology laboratory. You will be working with bacterial colonies. Just don't worry about this. I'm just trying to get you familiar with the whole idea of growing bacteria in the laboratory. So let's get back to this slide. I was telling you that we are able to classify bacteria based on their macroscopic and microscopic appearances. And also you can classify bacteria based on how do they look under the microscope. Just ignore the title of my slide. It should be microscopic distinction. There was a mix-up, okay? I will correct it and then send the PDF for you guys. Just ignore the title for now. So this is the microscopic distinction of bacteria. Some bacteria, as you can see, they are spherical. Some bacteria are rod-shaped. Some bacteria are curved. So different bacteria have different shapes, and we can, classify them based on how they look under the microscope. So we can classify bacteria based on their macroscopic and microscopic appearances. Also, we can classify bacteria based on their characteristic growth. You know, we can classify bacteria based on what requirements do they need to grow. Based on their, we can also classify bacteria based on their metabolic properties. For example, we can classify them based on, you know, if they need a certain sugar to grow or not. Also, we can classify bacteria based on their antigens. I don't know if you have studied the antigens in your immunology courses or not, but antigens are able to stimulate the immune system. Different bacteria have different antigens, and we can classify them based on their antigens. And finally, the most precise method to classify bacteria is based on their genetic material or proteins. So the most precise method for classifying bacteria is based on their genetic material. What types of genes do they have? What types of proteins do they have? So basically, this is the precise method for classifying bacteria. Do we classify based on turnover number too? You know, if you study different, microbiology books, maybe they use other criteria to classify bacteria. But based on the, medical microbiology, Murray's medical microbiology, which is the source that we are using to teach you, these are the only criteria that we use for classification of bacteria. But yeah, you can say that some bacteria they have a very short, generation time. Some bacteria have very long generation time. So it depends on again. It depends on the genes that bacteria have. Okay, so let's get back to the various shapes that bacteria have under the microscope. You know, as I said, different bacteria, they have different shapes, which help us to identify them when we see them under the microscope, which is very important. So if a bacterium is a spherical, it is called a coccus. You're seeing a coccus bacterium on the left side of this image. It is a coccus. It's spherical. It's called a coccus. And the plural form of coccus is cocci. So if you have more than just one coccus bacterium, we call them cocci. If a bacterium is rod-shaped, it is called a bacillus. And the plural form of bacillus is bacilli. So if you have more than just one bacillus, they are called bacilli. If the bacterium has a shape that is intermediate between a coccus and a bacillus, it is called a cocobacillus. And the plural form of cocobacillus is cocobacilli. When the bacterium is rod-shaped, but it has pointed ends, it is called a fusiform bacillus. The plural form of fusiform bacillus is fusiform bacilli. If the bacterium is rod-shaped, but it is curved or comma-shaped, it is called a vibrio. The plural form of vibrio is vibrios. If the bacterium is helical, but it is flexible and long, it is called a spirochete. As you can see, it is called a spirochete. And the plural form of a spirochete is called spirochetes. If the bacterium is helical, but it is short and rigid, it is called a spirillum. And so spirochetes are longer and more flexible spirilla, as I told you. So basically, these are the shapes that bacteria have when you see them under the microscope, and we can use these shapes to classify bacteria. So as you remember, I told you that the most precise method for classifying bacteria is by analysis of their genetic material or proteins. For genetic material analysis, we can use DNA hybridization, polymerase chain reaction or PCR amplification, and also DNA sequencing. So among these methods, PCR amplification is the cheaper one. So we have some nucleic acid amplification tests that we call them NAT or NAAT. And sometimes in order to identify bacteria, we use these types of tests, which are amplification tests. You can, amplify a specific gene in bacteria. If your bacterium has that specific gene, you can amplify that. And in that case, it means that your bacterium, yes, has that. So basically, there are specific genes that you can use in order to identify bacteria, and the method for identification of bacteria based on the smell sensation. So let's talk about bacterial cell structure. Now, here in this slide, you're seeing a bacterial cell. It's a bacterium. First of all, this bacterium has a cytoplasm. Cytoplasm is a gel-like substance which contains water and various molecules like, small molecules, macromolecules, other, cell components which are necessary for the cell function. Now, first of all, you're seeing the chromosome in the middle of the cell. So as I told you, bacteria, they don't have a nucleus, but they do have a chromosome. Most of the time, they have only one copy of the chromosome, which is circular. The area that you can find chromosome inside the cell is called nucleoid. They don't have a nucleus, but the area which you can find the chromosome inside the cell is called nucleoid. And this nucleoid is not membrane bound, as I told you. The cytoplasm is enclosed by cytoplasmic membrane. So we have cytoplasmic membrane. I told you that cytoplasmic membrane is very important for the cells. If you remove the cytoplasmic membrane, or if there is something wrong with the cytoplasmic membrane, the cell will be dead. And additionally, most bacteria have a cell wall which surrounds the cytoplasmic membrane, and it actually protects the cell. But again, you already know that not all bacteria they have cell wall, but most bacteria, yes, they do have cell wall. And as you can see, the cell that we have here, it is able to move because it has a flagellum. It has a flagellum, so it means that this bacterium is able to move. Also, as you can see, this bacterium has ribosomes, 70S ribosomes. You know that ribosomes are very important for the cell, and they are responsible for synthesizing proteins. So let's start talking about cytoplasmic membrane. You're seeing part of the cytoplasmic membrane here. Cytoplasmic membrane actually encloses the cytoplasm, separates the cytoplasm from the external environment, so it protects the cytoplasm. The cytoplasmic membrane is a phospholipid bilayer, and there are various proteins which are embedded into this phospholipid bilayer. Also, as I told you, electron transport chain, if it's present in a bacterial cell, it is located in the cytoplasmic membrane of bacteria. So again, not all bacteria, they have electron transport chain, but if they do have the electron transport chain, it is located in the cytoplasmic membrane. And the cytoplasmic membrane, it contains a variety of proteins, including enzymes. These enzymes are responsible for catalyzing metabolic reactions for the cells. Another group of proteins that you can find them in the cytoplasmic membranes are called ion pumps. As you can judge by their names. Also, we have transport proteins. They are responsible for moving substances across the cytoplasmic membrane of the cell. And as you already know, we don't have a straws in cytoplasmic membrane of bacteria, but there are exceptions. The exception is mycoplasma. I already told you. So about the cell wall in bacteria, um, I told you that most bacteria, they have the cell wall, which is a crucial structure, and the cell wall actually protects the cell. It's a rigid structure. It also gives the shape to bacterial cell, and it protects the bacteria from the environment. Most bacteria, they have cell wall, and based on the structure of the cell wall, we have two main categories of bacteria: gram-positive and gram-negative bacteria. I will be telling you about this naming. Why do we call them gram-positive and gram-negative? I will be telling you in a few minutes. Just for now, just know that we have two groups of bacteria, two major groups of bacteria based on their cell wall structure. We have gram-positive and gram-negative bacteria. So in this picture, you can see the differences between the cell wall of gram-positive and gram-negative bacteria. So let's start with gram-positive bacteria. They have a thick layer of peptidoglycan. I will be telling you about the structure of peptidoglycan. Peptidoglycan is a molecule that, as you can see, it's um, the major part of the cell wall in gram-positive bacteria. So we have a thick layer of peptidoglycan in the in the cell wall of gram-positive bacteria, which surrounds the cytoplasmic membrane. So it's located outside the cytoplasmic membrane, and it protects the cell. In contrast, in gram-negative bacteria, we have a thinner layer of peptidoglycan, as you can see. And this thin layer of peptidoglycan is surrounded with another membrane, which is called outer membrane. So basically, this thin layer of peptidoglycan in gram-negative bacteria is situated between two membranes: the inner membrane or the plasma membrane, and also the outer membrane. So this difference in the cell wall structure is key to distinguishing these two groups of bacteria. In gram-positive bacteria, the cell wall may also contain additional components such as teichoic acids. I will be telling you about the structure of teichoic acid, lipoteichoic acids, and also complex polysaccharides and proteins. Okay, so in a cell wall of gram-positive bacteria, there may be some other components. Here it is the structure of peptidoglycan. Peptidoglycan is composed of linear polysaccharide chains which are cross-linked by peptides. So in the polysaccharide chain, we have alternating repeats of two modified sugar. We have N-acetylmuramic acid, or NAM, which is shown as M here in this slide, in this image. And we have N-acetylglucosamine, or NAG, which is shown as G in this image. And as you can see, these chains are connected to each other. They are cross-linked by peptides, short chain of amino acids. So these short chains of amino acids are just connecting, are linking the chains of polysaccharide together. So we have this structure in the cell wall. In gram-positive bacteria, we have several layers of peptidoglycan, while in gram-negative bacteria, as you already know, we have this thin layer of peptidoglycan. I told you that the cell wall is a rigid, has a rigid structure. So it means that if you put pressure on the cell wall, it'll break. This is the real photo that a bacterium is broken. You're seeing a broken bacterium here. So consider the cell wall as a rigid structure, not a flexible one. So we have a broken bacterium here. So it shows that the cell wall of bacteria is a rigid structure, not a flexible one. And here in this slide, you can see the basic unit of peptidoglycan. So several of these basic units, they just connect to each other, and they form the peptidoglycan. So as I told you, in peptidoglycan we have polysaccharide chains, which are made up of alternating units of two modified sugars, an acetylmuramic acid and an acetylglucosamine. And these sugars are linked by a bond, and this bond is sensitive to lysozyme. Are you familiar with lysozyme? So the covalent bond that we have between an acetylmuramic acid and an acetylglucosamine is sensitive to lysozyme. It means that lysozyme is able to break down this bond. So what is lysozyme? No, it's not the organelle. The one that you're telling me, the organelle is lysosome. Exactly. So lysozyme is an enzyme which we have in body secretions, in tears, in saliva, and it protects us against bacteria. And now you know why. Because the lysozyme is able to break down the bond that we have between NAM or NAM and NAG. And by breaking down this bond, it is actually breaking down the whole structure of peptidoglycan. And I told you that peptidoglycan in the cell wall, it's a rigid structure, and if it's broken down, it means that the protection of the cell wall will be gone. The cell will be susceptible. But remember that gram-positive bacteria are more susceptible to peptidoglycan. Are more susceptible to lysozyme in comparison to gram-negative bacteria. I will be telling you why in a few minutes. But for now, just remember that gram-positive bacteria are more susceptible to lysozyme in comparison to gram-negative bacteria. So anyway, let's get back to the basic unit of peptidoglycan. So we have two modified sugars. They are connected to each other by a bond, which is susceptible to lysozyme. And we have this small chain of amino acid residues, which are connected to an acetylmuramic acid. So the first amino acid in this chain is called L-alanine. The second one is D-glutamic acid. The third one in gram-negative bacteria is diaminopyruvic acid, or DAP. But in gram-positive bacteria, we have L-lysine as the third amino acid residue. The fourth and the fifth amino acid residues are D-alanine. But remember that the last D-alanine just gets released when the cross-link is forming between two chains of polysaccharides in the peptidoglycan layer. So the last amino acid residue that we have in this short chain of amino acids, which is a D-alanine. This one just gets released when the cross-link is forming between layers of peptidoglycan. So here in this slide, you can see how the cross-link forms between layers of peptidoglycan. So this cross-linking is different in gram-positive and gram-negative bacteria. In gram-negative bacteria, the fourth amino acid residue in one chain forms a peptide bond with the third amino acid of another chain, which creates a cross-link. But in gram-positive bacteria, the cross-link is formed again between the third amino acid residue of one chain and the fourth amino acid residue of another one, but with an additional peptide bridge made up of a 5 glycine molecules. So we have this longer cross-link in gram-positive bacteria, in gram-negative bacteria. But remember that in both cases, the third and the fourth amino acid residues are responsible for forming this cross-link, for forming this peptide bond. But in gram-positive bacteria, this cross-link is longer, as you can see, because we have this peptide inner bridge in the cross-link. So in this slide, I'm going to tell you the reason for naming bacteria as gram-positive and gram-negative. So we named them so because of this staining technique, which is called gram staining technique. This technique was discovered for the first time by a researcher, which was named Mr. Gram. So that's why we call this technique, staining technique, as gram staining technique. So it doesn't refer to, it's not referring to, the weight or something. Gram here refers to the name of a researcher who discovered the staining technique for the first time. So this gram staining technique actually differentiates bacteria based on their cell wall properties. When we apply this staining technique, gram-positive bacteria, they become purple colored, while gram-negative bacteria become red. So when you see them under the microscope, you will be seeing purple colored bacteria. They are gram-positive. If you see red colored bacteria, it means that they are gram-negative bacteria. So the process of gram staining actually starts with applying a purple dye, which is called crystal violet. Crystal violet molecules, they enter into the cell wall and stain the bacteria purple. So the first step in gram staining technique is applying crystal violet, which is a purple colored dye. Both type of cells, both gram-positive and gram-negative bacteria, they become purple after the first step of this gram staining technique. Then we add iodine, grams iodine. Grams iodine basically forms a complex with the crystal violet molecules and help the crystal violet to trap in the cell wall of both gram-positive and gram-negative bacteria. So this grams iodine, it's not a dye, okay? It's just a reagent which forms a complex with crystal violet molecules and helps the crystal violet molecules to stay in the cell wall, just help the cell wall to retain the crystal violet molecules. And then we apply a decolorizer, which is alcohol or acetone or a mix of both. This decolorizer basically remove the outer membrane that we have in gram-negative bacteria, and then it's able to remove all the crystal violet molecules in the cell wall, because as you already know, we have this thin peptidoglycan layers in gram-negative bacteria. So that's why this decolorizer is able to remove all the crystal violet molecules from the cell wall. So at the end of step number 3, gram-negative bacteria become colorless. But gram-positive bacteria, as you already know, they have this thick layer of peptidoglycan, so it means that the colorizer is not able to remove crystal violet molecules from the peptidoglycan. So that's why at the end of step number 3, gram-positive bacteria stay purple. And then we apply a counter stain, which is called safranin. Safranin is a red colored stain. And gram-negative bacteria, which are colorless now, they actually uptake the safranin and they become red. While gram-positive bacteria, they are already purple colored, and that's why they stay purple. So at the end of this staining technique, gram-positive bacteria will be purple colored under the microscope, while gram-negative bacteria will be red. So that's how you can differentiate between gram-positive and gram-negative bacteria in the microbiology laboratory. So it is very important. I mean, this gram staining technique is one of the most important staining techniques that we have in the microbiology. Um, we have medications, we have antibiotics, which are able to cover only, gram-positive bacteria or only gram-negative bacteria. So this is very important for physicians to know that if the bacterium is gram-positive or gram-negative, because it can help with their decisions for the therapy that they want to start for their patients. So it is very important. And of course, we have medications which are able to cover both types of gram-positive and gram-negative bacteria. So if the physician is not sure that the infectious agent is a gram-positive or gram-negative bacterium, they start with those types of medications which are able to cover both types of bacteria. So anyway, you will be performing gram staining technique in your practical sessions. So based on the cell wall structure, bacteria can be categorized into 4 groups. 2 major groups are gram-positive and gram-negative bacteria. And also additionally, we have Mycobacterium species. Mycobacterium species, they have a lipid-rich and waxy cell wall. And because of these, lipids that they have in their cell wall, you can just stain them with gram staining technique, because the dyes cannot penetrate easily into the cell wall and stain the cell, the mycobacterium. So in that case, to identify them, you need to use other staining techniques. We have this acid-fast staining technique that we use in order to stain Mycobacterium species. We will be talking more about Mycobacterium species, which are one of the most important bacteria in the medical microbiology. And as I told you, we have Mycoplasma species. Mycoplasma species, they don't have a cell wall. And as I told you, they have estrals in their cytoplasmic membrane, which strengthen the cytoplasmic membrane. So now I'm going to talk to you about 2 components that we have only in gram-positive bacteria. So in gram-positive bacteria, we have teichoic acid and lipoteichoic acids in the cell wall. So it means that gram-negative bacteria, they don't have teichoic acid and lipoteichoic acid. Only gram-positive bacteria have these components. So teichoic acids, they are polymers made up of either glycerol phosphate or ribitol phosphates. So they are negatively charged. Take a look at the image that you see on the right side of the slide. This is the structure of teichoic acid. So we have a phosphate group, which is connected to a ribitol or glycerol. Then we have another phosphate group, and this pattern repeats. So as you can see, we have phosphate, then we have ribitol or glycerol, then again we have phosphate, and so on. So because of the phosphate groups, the teichoic acid and lipoteichoic acid, they are negatively charged because of the phosphate groups. And so it means that we have two types of teichoic acids. We have glycerol teichoic acids and ribitol teichoic acids. When teichoic acid is anchored in the plasma membrane, it is called lipoteichoic acid. If it is attached to the cell wall, it is simply called teichoic acid. And teichoic acid and lipoteichoic acid actually help bacteria to attach or adhere to the tissues that we have in the body. And lipoteichoic acid help strengthening the cell wall of bacteria. But both of lipoteichoic acid and teichoic acid, they help bacteria to attach to tissues that we have in the body. In gram-negative bacteria, I told you that in the cell wall we have a thin layer of peptidoglycan and we have this outer membrane. Outer membrane is an asymmetrical bilayer. In cytoplasmic membrane, we have this bilayer. It's symmetrical. We have two layers of phospholipids, but in outer membrane, the situation is a little bit different from what we have in plasma membrane. Again, we have two layers in outer membrane, but two layers aren't identical. I will be telling you why. And the inner layer is made up of regular phospholipids, similar to those that we have in the plasma membrane in gram-negative bacteria, but the outer layer contains a special molecule which is called lipopolysaccharide that I will be telling you more about its structure. So just now you know that why do we call outer membrane an asymmetrical bilayer, because the inner layer is composed of phospholipids and the outer layer is made up of a special molecule which is called lipopolysaccharide. So we have this space between the cytoplasmic membrane and the outer membrane in gram-negative bacteria. It is called the periplasmic space. It's not an empty space. It contains components of the transport system. Also, it contains hydrolytic enzymes and other types of enzymes. These hydrolytic enzymes are responsible for breaking down nutrients, and these broken down nutrients, which are small molecules, can penetrate easily into the cytoplasm. But large molecules, no, they can just penetrate easily from the plasma membrane. But the small molecules, yes, there are some transport molecules, transport proteins, which are responsible for transporting these smaller molecules, which are the result of breaking down the larger molecules. So in the periplasmic space, we have hydrolytic enzymes, other types of enzymes, and components of transport system. In outer membrane, we have small channels that we call them porins. Porins are small channels, and molecules, including the nutrients, they just pass through porins and get access to the inside of the outer membrane. So these nutrients, if they are very large molecules, like large proteins, large peptides, large carbohydrates, they get broken down by the help of the hydrolytic enzymes. And the smaller molecules then transported into the cytoplasm by the help of transport system. So here it is the structure of lipopolysaccharide, or LPS, which we have only in gram-negative bacteria. It is also known as endotoxin, and it is a very important molecule which can cause severe outcomes when it is released into the body because of the gram-negative infection. I will be telling you more about the consequences of releasing the endotoxin in the body in your future sessions. But now we're just learning the structure of lipopolysaccharide. So I told you that the outer membrane in gram-negative bacteria is an asymmetric bilayer. The inner or internal layer of lipopolysaccharide, as you can see in the left image, is made of regular phospholipids, but the outer layer or external layer of lipopolysaccharide, we have this special molecule, lipopolysaccharide, or LPS. LPS has three main parts. I don't want to go into the details. I'm just uh, trying to make sure that you know the basic structure of lipopolysaccharide. So the innermost part of lipopolysaccharide is lipid A. As you can see in this picture, it's called lipid A. It is made of disaccharide diphosphate group, which is connected to fatty acids. Then we have core polysaccharide. In core polysaccharide, we have sugar molecules. And then we have O antigen, which is the external part of lipopolysaccharide. And again, in O antigen, we have various sugar molecules. So this is the structure of lipopolysaccharide. Lipid A, core polysaccharide, and O antigen, but always remember that lipopolysaccharide is toxic because of its lipid A part. So lipid A part is the most important part. Some bacteria, instead of having lipopolysaccharide, they have the smaller version of lipopolysaccharide, which is called LOS, or lipooligosaccharide. And in this type of molecule, it lacks the O antigen. For example, we have Neisseria. Neisseria is a bacterial genus. Species of Neisseria, they don't have LPS. Instead of LPS, they have LOS. They are gram-negative bacteria, but instead of having LPS, they have this smaller version of LPS, which is called LOS. In gram-negative bacteria, we have secretion devices. There are 5 types of secretion devices in gram-negative bacteria. They are responsible for transferring, transporting molecules from the cytoplasm of the cell to the outside. Sometimes some special types of secretion devices can transport molecules directly from the cytoplasm of a bacterial cell to the cytoplasm of another cell. Type 3 and type 4 act this way. They are able to transport directly molecules directly from the cytoplasm of the bacterial cell to the cytoplasm of another cell. And the molecules which are transported by these secretion devices or secretion systems help bacteria to attach or adhere to the tissues in the body. And sometimes these molecules help bacteria to have this intracellular growth. Some bacteria, they are able to grow inside the cells that we have in the body. And as you can see, sometimes these molecules help bacteria to have their intracellular growth. So generally speaking, both membranes that we have in gram-negative bacteria, I mean inner membrane or cytoplasmic membrane, and outer membrane, they are traversed by these secretion devices. No, glycocalyx is something else. This is the glycocalyx. Some gram-positive bacteria and some gram-negative bacteria, they have capsule or slime layer or glycocalyx, which is the outermost layer of bacteria. Most capsules are made up of loose polysaccharide layers, but there are also polypeptide capsules. They are poorly antigenic. So it means that when they enter into the body, they are not able to stimulate the immune system very well. So it means that they are hiding themselves from the immune system. Also, they are anti-phagocytic. So it means that the capsule actually protects the bacteria from phagocytic cells, like macrophages, which are responsible for engulfing, eating bacteria. And also capsule, protects the bacterial cell and also helps bacteria to attach or adhere to the tissues that we have in the body. So capsules are very, very important part of the cell if it is present. Some bacteria use flagella to move and flagellum. First of all, just know that the plural form of flagellum is flagella, and as you already know, it is used by bacteria to move. And some gram-positive bacteria and some gram-negative bacteria, they are able to move and they use flagellum. There are other ways for bacteria to move, but flagellum is specially used by bacteria to move. It's a rope-like propeller, and flagellum is anchored in the cytoplasmic membrane of bacteria, and it is composed of protein subunits, which are called flagellin. Some bacteria, they don't have flagellum at all. They are not able to move. Some bacteria, they have only one flagellum. Some bacteria have two. Some bacteria have several types of flagella. And if flagellum is present, it helps bacteria to swim or move. So for swimming, bacteria use flagella. But there are other ways to move. As I told you, I don't want to go into the details, but for swimming, bacteria use flagella. If the bacterium is able to move, there are other ways that help bacteria to move too. And here you can see some bacteria with their flagella. Some of them are having several types of flagella, as you can see. Some have only few numbers of flagella. In some gram-positive and some gram-negative bacteria, we find structures which we call them fimbriae. So the plural form of Fimberia is called Fimberiae. Fimberia or Peles are present sometimes in gram-positive and gram-negative bacteria. The plural form of Peles is Peli. They are hair-like structures. They are made of proteins. The pelin is the name of the subunit which is responsible for forming the pellets. Fimberia or pellets is sometimes they help bacteria to attach or adhere to the tissues in the host. So that's why Fimberia or pellets are very important. We have this special type of peli, which are called F peli or sex peli. This special type of peli actually help bacteria to transfer their genetic material between each other. We will be talking more about this type of pellets in the future lecture. In the left image, you're seeing two bacteria which are connected to each other by a sex or F pellets. They are transferring genetic material. And in the right image, you're seeing a bacterium. You can just compare, Fimberia with flagella. As you can see, flagella, they are longer appendixes on the cell. But Fimberia, as I told you, they are hair-like structures. They are shorter in comparison to flagella. Some bacteria, and only gram-positive bacteria, they are able to form spores or endospores. So never gram-negative bacteria are able to form spores, only gram-positive bacteria and some genera of gram-positive bacteria are able to form. Members of the genera Bacillus and Clostridium, they are able to form spores or endospores. If a bacterium is able to form spores, it means that they are able to form this resistant structure. And in that case, these bacteria have two distinct states of living, a vegetative state and a dormant state or spore. So as I told you, spores are highly resistant structures. They can withstand extreme conditions in the environment, such as heat, chemical agents, UV radiations, even sometimes spores are able to stay in the environment for a decade and longer. So they are very, very resistant to harsh environmental conditions. So when the vegetative cell encounters some adverse conditions, especially when they face um, nutrient scarcity, they form spores. The spores can remain dormant for a long time, and if the conditions are suitable again for their growth, the spores just germinate and get back to the vegetative cell. It's important to know that spore formation is not a means for reproduction, because one vegetative cell forms one spore and one spore gives rise to one vegetative cell. So it's not a way of reproduction. Just pay attention to this fact. When we study the structure of spores, we see that they are very dehydrated and they have multiple protective layers. They have minimum concentration of proteins and ribosomes. It's really important to know that spores are not dead, but they have this very low metabolism, near to zero. Okay, they are not dead; they are alive, but they have this minimum, metabolism. That's why they have minimum concentration of proteins and ribosomes. They also contain a chromosome. Chromosome is very important. It contains the genes which are needed for the cell function. So that's why we have a copy of chromosome inside the spore. And also we have a high concentration of calcium, which is bound to this special molecule, which is called dipicolinic acid. This calcium, which is bound to dipicolinic acid, actually protects the spore from the UV radiation. So that's basically why the spore has this high concentration of calcium bound to dipicolinic acid. And here in this image, you're seeing various layers of spores. The innermost region of the spore is called the core. In core, we have the chromosome and also essential cytoplasmic components. And the core is surrounded with a membrane, which is called inner membrane. And then outside the inner membrane, we have germ cell wall. And the germ cell wall itself is surrounded with another layer, which is called peptidoglycan cortex. We have this modified peptidoglycan in this layer. This cortex is surrounded with another membrane, which is called outer membrane. Just don't mix up the outer membrane here in this spore with outer membrane that we have in gram-negative bacteria. As I told you, gram-negative bacteria never form spores. So the outer membrane that we have here has nothing to do with the outer membrane that we have in gram-negative bacteria. It is another membrane. In order to distinguish this membrane with the inner membrane, it is called outer membrane. So it doesn't contain LPS and everything that we said about outer membrane in gram-negative bacteria. Just make sure that you know the difference between outer membrane that we have here and the outer membrane that we have in gram-negative bacteria. So the outer membrane is surrounded with another layer, which is called coat, which is very important layer of spores. It is a keratin-like protein layer, and it protects the cell from chemical agents. So it is very important. This layer is very important to protect the spore from the chemical agents. And the outermost layer of spore is called exosporium, which is a thin protein layer. What does germ mean here? Germ refers to, germ refers to another name of, microorganisms. It's not usually used by microbiology books anymore, but germ here refers to the cell wall that this bacterium has. Okay, you can just simply say the cell wall. And here in this table, you can just compare the gram-positive and gram-negative bacteria. Basically, it's the summary of whatever we talked about. First of all, again, you know that gram-negative bacteria have an outer membrane, but gram-positive bacteria lack outer membrane. Both type of cells, they have peptidoglycan, but peptidoglycan is thin in gram-negative bacteria while it is thick in gram-positive bacteria. And we have lipopolysaccharide in gram-negative bacteria outer membrane. We don't have lipopolysaccharide in gram-positive bacteria. And it means that we have endotoxin in gram-negative bacteria while we don't have endotoxin in gram-positive bacteria because they don't have LPS. Teichoic acid sometimes is present in gram-positive bacteria. We don't have teichoic acid and lipoteichoic acid in gram-negative bacteria. Gram-positive bacteria, some of them are able to form spores or endospores, while gram-negative bacteria lack this ability. Some gram-positive bacteria and some gram-negative bacteria, they have capsule. Capsule protects the bacteria from the immune system and helps bacteria to attach to the tissues. And lysozyme, the enzyme that we have in tears and body secretions, lysozyme is effective on the bond which connects N-acetylmuramic acid and N-acetylglucosamine. Lysozyme, so this is called the lysozyme. So I told you that gram-positive bacteria are more susceptible to lysozyme in comparison to gram-negative bacteria. The reason is lysozyme is a large molecule and it's not able to penetrate easily into the porins that we have in gram-negative bacteria. So if you remember, I told you that in gram-negative bacteria we have outer membrane, and outer membrane is impermeable. So it means that there are some, channels, tiny channels in the outer membrane, and molecules, if they want to enter into the cell, they need to pass through the porins. But lysozyme and other large molecules, they can just penetrate to the cell through porins. Even there are some large antibiotics that and they can just penetrate into the gram-negative bacteria because they can just pass through porins. And that's why gram-negative bacteria are resistant to those types of antibiotics or lysozyme. And gram-positive bacteria, they are more susceptible to the activity of penicillin. Penicillin is a type of antibiotic. You will be learning completely about different types of antibiotics, including penicillin. Penicillin is a type of antibiotic which interferes with the synthesis of peptidoglycan layer in bacteria. And as we have a thick layer of peptidoglycan in gram-positive bacteria, they are more susceptible to the antibacterial activity of penicillin. But gram-negative bacteria are more resistant. And as gram-positive bacteria, they have this thick layer of peptidoglycan. They are more resistant to drying and physical disruption in comparison to gram-negative bacteria. And some gram-positive bacteria and some gram-negative bacteria, they are able to produce exotoxins, which are some molecules which can have adverse effects on the host. And I will be telling you more about the exotoxins. For now, just remember that some gram-positive and some gram-negative bacteria, they are able to form exotoxins. Okay. Do you have any questions? Okay, so if you don't have any questions, let's take a break and I'll see you in 30 minutes.